I'll get started now. Sorry, guys, that was
Obnoxious.
a long delay. Did everyone sign in? Okay, cool. So this week is Renal Part 2, but I'm going to focus on Osmoconcentration. This corresponds to pretty much the first lecture video. It's only 3 short week. So I'll get into it. Office hours are tomorrow. Come by. There's my email. Yeah. When's the practical? Is that next week?
Yeah.
Okay. So you can use my study guide is up reminder. Okay. So zooming out before we start talking about osmoconcentration, this unit specifically is now talking about how we balance the contents of blood and urine. So this directly correlates to blood pressure. If you remember from last week, we talked about how more sodium is absorbed, more water is absorbed, which increases blood pressure. And then before we keep talking about this, I just wanted to throw this on the slide in case you confuse these terms. Solute refers to like something like ions or some other component dissolved in like a liquid or a solvent, and the solvent is like the liquid. So, because we're going to talk a lot about solute concentrations. So concentrating the urine. You can look at this in 2 different ways almost. So you can look at it as if I'm talking about if a person is overhydrated, then their urine's going to be hypotonic to the plasma. But then there are also some like animals that tend to have overhydrated urine. So that's kind of confusing, so I just wanted to point that out. But normal urine is isotonic to the plasma, meaning that the ion concentration of the urine and the blood plasma are the same. And this is like a medium-sized nephron. I'll have a visual of this soon. Overhydration is when urine is hypotonic to the plasma. Sea animals tend to have a lot of these cortical nephrons. They have a small loop of Henle, so less water is drawn out of the urine. The urine contains a lot of water. Dehydration is when you have hypertonic urine. So desert animals tend to have a lot of these juxtamedullary nephrons, which have a long loop of Henle, which allows more water to be drawn out of the urine and reabsorbed into the blood. For example, like desert animals may have a lot of these juxtamedullary nephrons, but when you're in the dehydrated state, you're also going to want to absorb as much water back from the filtrate as you can. So that's why humans tend to be lumped into that category, because we tend to be dehydrated. So this is a visual that I drew to help you kind of put like hypertonic, hypotonic into context. So hypotonic urine is when you're overhydrated. So inside the circle is the urine, like as if you're looking at a cross section of the nephron. So like this is the filtrate or the urine, and then outside is the blood. Hypotonic urine, there's a low solute concentration. Hypotonic, lower solute concentration of urine. Hypertonic, higher solute concentration in the urine. So That's why, like, when you're dehydrated, like, your pee might be, like, more yellow. It's hypertonic. Does that make sense? Or does anyone have any questions? Okay. So now we're going to start talking about osmoconcentration. This is done in humans to try to conserve or reabsorb as much water as you can because we're typically dehydrated. And this is done with a vertical osmotic gradient in the kidney. So, as you can see at the bottom of the screen, I'm going to blow that up in a second. As the nephron gets deeper into the medulla of the kidney, the concentration of solute, or the concentration, just increases, which helps draw more water out of the urine into the blood. So the cortex is the outside that has a concentration of about 300, I think it's milliosmoles is the unit, I'm not sure. And then the outer medulla is 400 to 600, and the inner medulla is 900 to 1200. Basically, it's increasing as you go deeper into the kidney. And this helps draw more water out of the filtrate and into the blood. Because water is drawn towards a higher solute concentration. So here's the figure at the bottom of the last slide. You can just see that as you get further into the medulla, there's a higher concentration of solute. And then there are three main contributors to maintaining this concentration gradient, or to helping concentrate the urine. The first is the salt story, then the urea story, then counter current exchange. And we're gonna draw a picture of each of these to understand the differences and how it helps concentrate the urine. So first, here's the salt story. This one's known as functional coupling. And it's basically because the descending and the ascending loops of Henle are so close together that they kind of work together to concentrate the urine. On the ascending side, we're going to draw this in a second, but there's active transport of sodium out into the blood. And then that active transport of sodium out allows water from the descending loop to come out, which will make more sense in a second, which helps concentrate the urine. This is also a form of a countercurrent multiplier, which means that we're helping the concentration gradient be maintained in the medulla. We'll talk about that more later. Okay, so I'm gonna draw it and then I'll put it on the. Does anyone have more dry erase markers? You do? Okay, because I only have two colors and that might just be, I don't know where my other ones went. I'll start with this one. Oh, that's okay. That's okay. Don't worry about it. We can use red and green. We can rock with it. Okay, so I'll start with the salt story.
I have some.
Oh, you have some?
Yeah.
Cool. Thank you.
You know, mine is like double with black and green.
Oh, awesome. Oh, perfect. Thank you. Okay. So the first story that helps us concentrate the urine is salt, the salt story. And then I'm just going to draw the nephron as good as I can. Okay. So this is the descending limb, thin ascending, thick ascending collecting duct. Okay. I'll actually label it so we have descending. And collecting duct. So in the ascending limb or loop, there are active transporters that actively pump sodium out into the blood. Actively pushing sodium out. You guys remember that from last week that water follows sodium? So then in the descending limb over here, water is going to be drawn out. So that's basically all of that saying. We have active transporters in the ascending limb that help take sodium out of the filtrate into the blood. And then that draws more water out of the descending limb. Taking water out of the filtrate is concentrating the urine. And then just for context, this is just the direction of the filtrate. And this is due to the close association between the descending and the ascending loop, and that's also known as functional coupling. So you're coupling these two units. Functional coupling. So here's that on the slide. Is that confusing, or what do you guys think? You guys got it? Okay, cool. Move on to the next one. Urea story. So now urea in the filtrate is recycled to make a concentration gradient to draw water into the blood in the presence of ADH. So basically there's urea in the filtrate, and similar to sodium, water follows the urea. So if we take urea out of the filtrate, we're going to take water out of the filtrate into the blood. So I think it's just will make more sense when I draw it again. Do you guys want me to do 3 separate drawings, or should I just put them all on here? 3 separate? Okay. I feel like that might make more sense. 2, this one orange. Urea. So now you can basically just copy paste nephron. But here we're just going to focus on the collecting duct because the collecting duct is permeable to urea. So urea is in the filtrate, like everywhere, it's not just there. But we're permeable to urea in the collecting duct, so urea will come out. And water will follow. Because water goes from a lower to a higher solute concentration. So it's following the solute. And taking water out of the filtrate concentrates the urine. So we're concentrating. So urea leaving the filtrate increases its concentration in the medulla, which increases the concentration gradient, allowing more water to leave. Yes. And that contributes to the countercurrent multiplier, which refers to that osmoconcentration gradient in the medulla. Does that make sense? Any questions on that urea story? Okay, now the last one is countercurrent exchange. And the purpose of this one is to maintain the countercurrent multiplier. Is everyone good on what countercurrent multiplier is referring to? Okay, so countercurrent multiplier is basically just referring to the concentration gradient, this concentration gradient in the medulla. So countercurrent multiplier is maintaining that like higher concentration of solutes as we go deeper into the medulla, because as you go deeper into the medulla, then it allows water to come out. So here we're maintaining that countercurrent multiplier or concentration gradient, the purple, by drawing urea out. That increases the solute concentration, increases the countercurrent multiplier. Here we're contributing to the countercurrent multiplier by taking sodium out of the filtrate. Increases the purple, increases the countercurrent multiplier, increases the concentration gradient. So now with countercurrent exchange, we're trying to maintain that gradient. So again, there's like a definition for countercurrent multiplier referring to the osmoconcentration gradient. Countercurrent exchange now is referring to the opposition of flow that maintains the gradient. So this one can be a little confusing, so we're going to draw it to explain it. So 3, this one green. Counter current exchange. I'm going to draw the nephron again. Okay, so we have the nephron drawn, but this time I'm going to include the capillary or the blood source that's closely associated with the nephron. So I'm going to draw a blood vessel. And this blood vessel that's specifically associated closely with the nephron is known as the vasa recta. So counter current just means that, so we know that the filtrate is flowing in this direction through the nephron, down the descending, ascending to collecting duct. Counter current refers to the opposition of flow, so the blood in the vasa recta is flowing in the opposite direction. And this is important in maintaining that concentration gradient that we made through these mechanisms. So going back when we talked about the salt story, we know that there's active transport of NaCl out in the ascending limb into the blood. And that active transport of NaCl out, so NaCl is coming out, traveling up here, and then that helps draw water into the blood. So then water is drawn out into the blood there. Then now, so we just looked at it from the perspective of the blood, increasing NaCl in the blood. When it travels back up here, then water is going to be drawn into the blood. If you look at it from the filtrate, leaving water leaving the filtrate in the descending limb is going to concentrate the urine here, which then allows NaCl to come out here. So it's kind of, the system works together when you have the opposition of flow in countercurrent exchange. Does that make sense so far? Okay, something else to add here is the recycling of urea. So since the collecting duct, now we're talking about the urea story, the collecting duct is permeable to urea, so urea leaves, urea, some enters the blood, comes down, and then there is a transporter for urea. I believe it's in the descending limb. So it's coming through the blood, traveling, and being recycled back into the filtrate here. So that like maintains a cycle. So urea doesn't just keep leaving into the medulla, enters the blood, and comes back into the nephron in the descending limb. So it can be maintained in the filtrate. I'll come back to that. So this is what I've talked about so far. Any questions on countercurrent exchange so far? Okay, I'm just going to draw what concurrent exchange would look like, which is if the blood and the filtrate were flowing in the same direction. So maybe it can help make more sense of why this is necessary to recycle and maintain the gradient in the medulla. So this would just be concurrent flow. And this is not this is not correct. So filtrate would flow in this direction. And then the blood would also flow next to it. So first of all, looking at urea is only permeable in the collecting duct. So there's no recycling of urea in concurrent flow. It's not being carried back into the filtrate so it can cycle back. It's just leaving and it's never going to be replenished in the filtrate. Talking about the salt story, NaCl is actively transported out of the ascending limb. The NaCl is going to be carried this way, away from the descending limb, instead of being carried this way and helping water be drawn up here. So this is what I just said, written down when you review. And here's the concurrent exchange. You'd lose the concentration gradient. Okay, so now I just wanted to return to the first slide that we talked about and relate it to the length of the loop of Henle. So the length of this region right here differs based off of like how much you're trying to concentrate the urine. If you're trying to concentrate the urine more, then there's a longer loop because it goes further into the medulla and there's a higher concentration gradient. It pulls more water out. And those are the juxtamedullary nephrons. And the opposite is true if you don't need to pull as much water out of the urine. Those are cortical nephrons. And here's a representation of the length of the loop of Henle in relation to the concentration of the urine. Does that make sense, or do you want to draw this out too? What do you guys think? You got it? Okay. Nice. And then this is just a different way to look at it. You can see that the juxtamedullary nephron goes further into the medulla. They're longer, and they're able to draw more water out, concentrate the urine better. Okay, so now I've got some top hat questions. Which region of the nephron is permeable to urea? Does anyone want to walk me through what they're thinking for this one?
I said that it was the urea is permeable to the collecting duct
Mm-hmm.
because urea is exiting out and then enters the blood and then would get reabsorbed back in the descending route to help keep that concentration gradient.
Yeah, exactly. Perfect. Nice.
Would that mean it's also permeable descending independently?
No. So it's only permeable in the collecting duct. That was a good question. There's a transporter or
Okay.
channel or something.
So it needs help from some. It doesn't need like a protein to leave the collecting duct.
Yes.
It needs it to enter back in.
Exactly. So it can go right through in the collecting duct, but it needs, I think it's a channel, but some sort of protein or something to help it enter the filtrate in the descending loop. Sodium leaves the nephron to enter the blood in the blank via blank. We'll say specifically talking about the salt story for that one. You guys got it. So it's active transport in the salt story. So you got sodium out of the filtrate, and it's in the ascending line. Desert animals would have a high concentration of what type of nephron? Exactly. So juxtamedullary go further into the medulla, where there's a higher concentration gradient, which helps draw even more water out of the urine into the blood. So you're conserving water. countercurrent exchange refers to? Yes. So it's the opposition of flow, it's countercurrent, and it's between the filtrate and the nephron in the blood. The purpose of countercurrent exchange is to? Yes, so countercurrent exchange helps maintain the countercurrent multiplier, the concentration gradient in the medulla. It helps recycle urea and concentrate the urine. Okay, so I didn't talk about this. I think this is in reference to the second lecture video, but I wanted to throw it on there. The external urethral sphincter is what muscle and under what control? Nice. Skeletal involuntary control. Things that are skeletal muscle tend to be under voluntary control, whereas things that are smooth muscle tend to be under autonomic or involuntary control. What three systems or organs play a role in maintaining fluid homeostasis? I didn't talk about this also. Can you select multiple? Okay, well, I didn't know how to change the settings this time. They switched around top hat, but we can talk about it. Look at that, we got all three. So kidneys, hypothalamus, and cardiovascular system. Perfect. Okay, so that's all I have for you guys today, but feel free to stay back if you have any questions or come to office hours.